over to Nicholas to start.
Okay, so, it's a great pleasure today to have Brian Bender as our speaker. Brian is a true expert in G-protein coupled receptors, which are of great interest as pharmacological targets in the pharma industry. So Brian is currently a lead scientist at Heptares, where he has been for the last 2 years. But it turns out that Heptares was nervous about Brian talking about his current industrial work. So today's presentation will mostly focus on, will focus on the work that Brian did before he joined the industry. But fear not, this is a work of great interest, and indeed, Brian, in fact, started in science as a trained structural biologist. He's trained in crystallography during his undergrad studies, and then he moved for his PhD to Vanderbilt University, where he developed methods for better prediction of GPCR structures. And then for his postdoc, he went to work with Brian Choquette at the University of California at San Francisco. Brian Choquette is, of course, very well known for virtual screening. And that's in part what Brian did there, although he did virtual screening on very large database of compounds. This coupled with modeling of GPCRs for which the structures are not known. So one of the aims was to test how model structures are useful for these virtual screenings. Brian has also some interest in pharmacology. So he has a very deep and long-running experience in GPCRs and work very relevant to industry and drug discovery. So I'm really looking forward to this talk, and the floor is yours, Brian.
Excellent. Thank you, Nicholas. That was a very kind introduction, and thank you to all of CCPBioSim for welcoming me here today to present. Let me get the slides up and running for all of you. Okay, and you're all seeing this? Good.
Yes, I can see them.
So yes, I do have to give an apology for those of you that were hoping to hear about AlphaFold today. I won't be able to present on that, but I am presenting on still quite new research coming out of my postdoc. Only some stuff published just in the last week, so it should still be, I hope, a very interesting talk for those of you that stick through to the end. My focus, as Nicholas indicated, is on G-protein coupled receptors, otherwise known as GPCRs, but most of what I'm talking about today can be applied to many other classes of proteins. So to get started with G-protein coupled receptors, for those of you that are unfamiliar with this family of proteins, they are proteins that sit in the cell membrane. They're in fact the largest family of membrane proteins in the human genome, and their primary role in cells is actually to signal to cells what's going on outside of them. So they respond to stimuli coming from the extracellular milieu that then come and bind to the receptor, and in this binding process activate the receptor and allow it to then couple to downstream signaling pathways to tell the cell to modify its activity based off of what's going on around it. This process of going from a just sort of basal state in the receptor into an activated state is coupled with a series of conformational changes that structural biology is now starting to get a handle on and understand the fine mechanism of activation. GPCRs are one of the in my opinion, most pharmacologically targeted families of proteins, most relevant family of proteins. No, I mean, they're up there with kinases and ion channels in particular as the most heavily targeted proteins, in part because of the way that they act. You know, they sit on the cell surface and they respond to extracellular stimuli, and that extracellular stimuli can be both endogenous hormones, but exogenous small molecule and peptide biologics as well. This review is now getting a bit old, saying 50% of drugs are GPCR based. It's more like 30% now, given the rise of other classes of drugs. But in fact, many well-known compounds activate GPCRs. Even if you don't know what a GPCR is, I can guarantee you've heard of at least one of these compounds listed here that activate receptors. So caffeine, if you drink coffee, melatonin, endorphins, adrenaline. Actually, light, it's not a compound, it's a photon of light can activate the GPCRs that are responsible for vision in our eyes. Dopamine, serotonin, histamine, and of course, some illicit substances have taken advantage of the same signaling pathways as some more beneficial compounds. Now, I did say GPCRs, one of the largest families of membrane proteins. In fact, they're highly diverse. They've evolved for a long time, and as such have become quite distinct individual receptors. And if you plot all of, and what I did is I just did a percent identity matrix of all GPCRs against all other GPCRs, they can be grouped into certain classes, A, B, C, and F, based off of their extracellular domains. But even within a given class of GPCRs, they're still quite dissimilar from each other. There's a few clusters that you can see, these little islands of blue, but largely the identity between any given receptor is quite low. In fact, between all GPCRs, the percent identity is only 18%. And that only rises to around in the mid-20s within any one of these families. Despite this very broad difference in sequences, they all share a similar 3D topology. So these GPCRs are also sometimes known as 7-TM or transmembrane receptors, and that's because they are comprised of 7 transmembrane helices, the N-terminus sitting in the extracellular space and the C-terminus sitting in the intracellular space. And as more structures have become available of GPCRs, regardless of the class A, B, C, or F that I just mentioned, they all adopt the same topology when you align them on top of each other in the transmembrane domain. And this is one thing that's quite interesting and unique about GPCRs, that despite the broad sequence difference, the 3D shape is the same. And GPCRs for a long time were sort of intractable to structure determination methods. Brian Kabilka started working in the 80s trying to get a crystal structure of a GPCR, and it wasn't until 2007 that he finally succeeded. Lots of failed grants went into that first structure, but he got that first structure. And in the last 20 years, as you can see, the number of structures of GPCRs, of unique receptors, has only grown exponentially. And significantly in the last 5 years, it's the rise of cryo-EM that's really allowed us to continue to push into this field of novel receptors. And so at the moment here in 2024, we're quite fortunate. We have just about 200 structures of GPCRs determined, and they cover a wide variety of the families of GPCRs. So all the class A or rhodopsin-like families, there's high coverage of receptor structures there, as well as dotted throughout the adhesion, secretin, glutamate, and frizzled. Not so much over here in the frizzled taste receptors. But even with 200 structures, you might think this is we're getting near to the end, and in fact, that's not the case. If I just plot on however remaining GPCRs there are without structures, it's an equal number. So there's 800 GPCRs in the human genome, 400 of which are considered pharmacologically relevant. And so 400 divided by 200, we have as many unknown receptors without structures as we have known receptors with structures. So there still remains a need to both determine structures experimentally, but have methods in place that allow us to predict those structures in the absence. So some of the tools that are needed for GPCR structure prediction is, of course, how to understand how current drugs are binding. So we have, as I mentioned, many current FDA-approved drugs targeting GPCRs, but we don't have experimental structures of them all bound at the receptors, and the ability to continue to improve on them and develop fast followers, for example, relies on having accurate structures. Of course, wanting to predict new drugs binding to a given receptor, and then understanding signal transduction, conformational dynamics, and disease mutations. And I wrote disease mutations, but actually, as we learn more about the human genome through the 1,000 Genome Sequencing Project, we see instances of polymorphisms that are present throughout the human population. And these polymorphisms actually account for different reactions to the pharmacology of various GPCR drugs. One drug acting at one person might behave differently at another person due to polymorphism. So structural knowledge allows us to start teasing this apart and developing more specific medicine for patients. So the purpose of the talk today really is going to be focusing here on this second point, predicting the receptor structures to allow for new drug binding. So where this all started for me, at least, was with the software known as Rosetta, which is really a combination of various bits of code that do different tasks. And by combining all of them together, you can build up very complex models. And Rosetta is starting, well, I mean, it's been well known for a long time due to David Baker and his all the novel proteins that he's generating in his lab, both vaccine designs, but novel functional protein origami and so on. But I was focused not necessarily on generating new proteins, but understanding how at the time do we get structures of proteins that already exist. And Rosetta is well suited for folding de novo proteins just from the sequence alone. When I started this project in the mid-2010s, it was quite good at taking small globular soluble proteins and folding them up here like the T4 lysozyme, or very simple membrane proteins comprised of straight alpha helices that pack quite nicely against each other. However, when I wanted to use it to predict a structure of a GPCR, in this case rhodopsin, I got a very wrong answer. I mean, just a complete jumble, a mess compared to what the true experimental structure of rhodopsin was. And this is just because the receptor sequence, it was getting quite long. It was 300 to 400 amino acids, which was really pushing the limits of de novo structure prediction. And the arrangement of the helices within the membrane, though having a defined topology, now that we've have the structures, getting all the long range contacts between those helices correct is quite a challenge. So in the absence of accurate de novo folding, and again, in the absence of AlphaFold, because that was not present at the start of my PhD as much as I would have loved that, the best next case scenario that you could use is comparative modeling or homology modeling. With homology modeling, you take a related experimental structure, so a protein that has some degree of similarity to the protein that you want to predict. You thread over that structure your target sequence. You get something that looks maybe a bit disconnected because not everything aligns perfectly between the target and the template. Through a process of energy minimization and any loop building as needed, you get an output model, and this is your homology model. This is the very basics of homology modeling that's been used for now decades. And this simple method of going from threading your target sequence onto a template has one significant limitation, and that is you have to have a very good template in order to get a good model. there's almost a linear correlation between the starting template accuracy and the model accuracy of your target. So the higher the percent identity of your template to your target, the more likely you're going to have an accurate model. But as I said at the beginning of this talk about GPCRs, the average sequence identity of any given template for GPCR for any other GPCR is around 20%. And so that template accuracy is going to be quite low. Therefore, your model accuracy applying this method of single template threading onto a GPCR is going to result in very poor models. So we don't have a good method for de novo structure prediction, and we don't have a good method for homology modeling. What next? In the early 2010s, there was a method from Rosetta put out that allowed for the use of multiple templates during the homology modeling process. And the reason for this was that a given region of your target might have high similarity to one template, but very low similarity in a different region, but that other region has high similarity to another template. And so by allowing the method to mix and match between its various templates, it could actually find what is the most appropriate template for the local sequence of your protein of interest. And using this method now, there was no longer a linear relationship between the template sequence identity and the model accuracy. And in fact, there was high accuracy for any model generated with a template down to about 40% identity. Once you're below 40% identity, you have the possibility of getting a highly accurate model, but you also have probably an equal probability of getting a very poor model. And knowing a priori if you're going to have a useful model or not is impossible in this lower than 40% sequence identity range. But again, this is where we are with GPCRs, the low 20%, 30% sequence identities. This didn't stop me at the time, thankfully. I decided to push on with something that probably was likely to fail. And through a lot of tedious hours in the lab, I found a way forward. The first is that I was really benefited by the continued availability of GPCR structures. At the time, there was probably a new structure every couple of months, and each new structure was really enlightening. Now we have dozens of structures every week of GPCRs. It's really exhilarating, honestly, in this field at this time. But with each new structure, we started learning sort of the various intricacies of the different receptors with respect to one another. And the first thing that really came out was that there had been a method for aligning GPCR sequences based primarily on conserved residues within each of the transmembrane helices. And you would generate a homology model based off of using those single conserved residues and propagating your sequence alignments based around it. But as more structures came out, it was evidence that there were defined gaps in these sequence alignments that were family specific. And so now we had this understanding that if you're modeling a class A family gamma receptor, you need to include a certain break in your sequence alignment with respect to other templates in order to maintain the right helicity in the model that you're generating. And doing fixes like this and others, we're able to correct really bad basic alignments that in the end would not result in continuous helices, with now much better looking helices that are positioning side chains correctly for the interior versus exterior of the receptor. And so using some of these optimized sequences, I compared how the receptor prediction accuracy was with other automated methods. Some of these methods are structure-based alignments, some of them are sequence-based alignment. I was really using a blend in the end of sequence and structure-based knowledge. And this is showing the change in the RMSD over a benchmark of proteins that I predicted And it found that in every single case of a different sequence alignment compared to the one that I generated, I was getting worse RMSDs compared to the method developed. This was found both within the transmembrane region as well as the full model. And I called out specifically extracellular loop 2 of GPCRs because this tends to be the longest and most complex loop of all GPCRs, and therefore the one that's most difficult to predict. And very nicely, I was able to overcome significant RMSD errors from certain alignment methods using the new blend of sequence and structure alignment. And then the other really nice surprise that I started seeing as I was going through my various case examples is that if I started removing templates at various levels of percent identity, so first only using templates between 30 and 39, so below that sub 40% threshold, and then going lower and lower and lower, how low could I go with a percent identity of a template to my given target and still maintain accuracy? And what I was finding over and over again by using improvements in sequence alignment and other changes to the code, I apologize, it's a very abbreviated version of all the work that I put into it, but I was able to then get highly accurate models with templates down to 20% identity. So we went from needing to have models above or templates above 40% to now needing them as low as 20%. And what does that really mean for going out and predicting structures? So this here again is that same figure of green are the receptors that have structures, red are the receptors that don't have structures. And this is plotting now what is the best template for any given GPCR with the available structures that we have. And yes, 200 of these now have a structure, and so therefore we probably aren't building a homology model of them. But then another quarter of them have already a template above that 40% threshold. So they would have been fine in the previous method without any optimization, and most likely have given a good model. But then there's another quarter of the model of the receptors that have sequence identities in the 30 to 40% range or the 20 to 30% range. And then finally, just 10 receptors currently that their best template is only less than 20%. And so with the previous method requiring the templates of 40%, we could only accurately model at this given present moment 3/4 of all receptors, including the ones that have structures. But with the new one, we now can get that down to all but 10 out of the 400, which would significantly change the landscape of receptors that have either experimental structures or models with highly accurate predictions. Of course, this is all changed with the advent of AlphaFold, and AlphaFold can do a lot of this very, very accurately and also very quickly compared to the amount of time and effort that went into the homology modeling process. But I still, to this day, I use both equally, and I find valid reasons to use both depending on the application at hand. So this here gets us a structure of a GPCR, which is really nice. I mean, it's really helpful just to understand how are residues laying in the transmembrane region. If you already know of mutations that affect signaling, you can start plotting them on here. You might look to see where my pockets exist that ligands could bind into. But this is really a static image. It's a single structure, a single model of an experimental state. And so the first, next logical extension is what's the inactive versus the active state conformation. And methods with AlphaFold in particular are really pushing the envelope of what we can predict accurately for GPCRs in active and active state. And if you are interested in accurate models of GPCRs in either of these states, I do suggest you look at the GPCR-DB, which is organized by David Gloriam, that has now a collection of AlphaFold models in these various states. But again, this only represents a snapshot of the receptor. And in particular, it doesn't say anything about what these receptors are interacting with. They don't really exist without partners in the cell environment, and it's important that they have binding partners externally and internally in order to convey signals. So, the next question is, what do endogenous ligands look like? Small molecules, peptides, and proteins, and of course, what do exogenous ligands, such as drug compounds, look like at the GPCRs? What do they look like in their signaling complexes with G proteins and arrestins? Some GPCRs don't exist as monomers. Some of them exist as dimers. Some of these dimers are constitutive. Some of them are transient. So getting methods to predict all of these accurately is quite important. But then also what these models don't really capture is what's happening in the dynamics of the N and the C termini, as well as if there are additional domains, particularly in the extracellular space, that are important for signaling events in the GPCR. Again, AlphaFold is really starting to tackle many of these issues. What does the complexes look like with G protein and arrestin? AlphaFold will give you models of the N and the C termini, but if they're disordered, they will be disordered. And also what's becoming really nicely is starting to stitch together the domains, the extracellular domains of GPCRs, so the relative domain orientations are often incorrectly predicted. And what I want to focus on now, of all the potential things that we could model, now that we have better modeling methods, is particularly small molecules and peptides at the GPCRs. So one of the first things that I was given after I developed my method was, how do you predict the structure of a peptide ligand, in this case ghrelin, bound to its receptor? And so I was given already an NMR ensemble of ghrelin bound to membrane, and then I had my APO models of the ghrelin receptor. And hopefully I can convey to you that between the conformational dynamics of the peptide alone in the unbound state and the receptor in the unbound state with all of its loops, there's a significant conformational dynamics at play, and trying to basically get one ball of spaghetti to bind to another ball of spaghetti is a challenging thing to predict. And I needed to come up with a new way of how do I take this output from Rosetta that I had previously generated and make this meaningful to allow a peptide to bind. And so at the time when I was working on this, my thought was actually, if there's a lot of dynamics here in the loop regions, and there is some structural convergence here in the helices, let's just ignore all of the gross changes, the loops, and focus on the part of the model that's really well converged. And so that's what I did. I cut out all of the highly dynamic region and focused on the transmembrane region. This is the part that was well converged in all of my models. And then I docked just a part of the peptide. So just 7 residues of the full ghrelin peptide. And I generated a set of models of this small peptide bound to this loopless GPCR. I was then able to start building up the helices and the loops in the presence of that docked peptide. I continued to extend the peptide, extend the loops, and so on and so forth until I had a complete model. And in the end, I went from these 2 balls of spaghetti through this iterative docking and rebuilding protocol to a very highly converged model for both the peptide ligand as well as the extracellular loops of the receptor. And this is something that we had observed ourselves and many other people had observed, is that the binding pocket, when you predict the binding pocket, it's very influenced by the presence or absence of the ligand. And in fact, the absence of a ligand in the binding pocket during a structure prediction has very negative effects. Essentially, the methods, they see a void, an open space in the structure, and that's energetically unfavorable. And so the energy minimization protocol is trying to collapse that pocket, and it does it either by positioning side chains of the binding pocket towards each other so that that pocket collapses, or even in the worst case, moves the backbones of the binding pocket closer together to close that void. And so we really needed this iterative process to keep the pocket open while we continued to build up the receptor and the peptide together to get this highly converged state. And it was nice then to get NMR data back on the bound state, showing that going from the unbound state where the primary secondary structure is random coil, and that's exactly what we see, that in the bound state becomes primarily alpha helical, particularly after this residue 7. We see that captured in the models that were generated by the method. So it's disordered coil in the N terminus that becomes a helix in the C terminus of the peptide. And this was a very complex and highly intervened process. I had to keep making decision points throughout in order to get this model, but it was the best that we could do at the time. And again, AlphaFold, it can do this in the matter of hours and give very highly accurate peptide binding states for proteins. That is both beautiful to watch, but also incredibly frustrating after having put in the amount of time and effort that I did to do this in the pre-AlphaFold days. So that's peptides, and peptide binding, as I just mentioned, is in part, I would say, solved by the development of AlphaFold. But how do we move into something a bit more interesting, more complex, I should say? And this is the case of small molecules. So the reason AlphaFold does well with peptides is because they're the same polymer. So both the target receptor is comprised of amino acids and the ligand peptide is comprised of amino acids. So it can use the same force field to co-fold each other together. You don't necessarily have that when you have a small molecule, because you have any sort of ligand geometry and atom combinations that can't be defined by the 20 standard amino acids. And so at the time that I was interested in trying to tackle this question, how do we optimize our models for small molecule docking, I was starting to work on a family of proteins called the RF amide receptors. There's 5 RF amide receptors here circled in black that are activated by a series of endogenous peptides here with the various colors. And the real challenge to studying the biology of this family of receptors is that the endogenous peptides are highly non-selective. I've mapped the efficacy of the various peptides for the various other receptors, and you can see this interwoven complex of peptide receptor combinations. And so if you try to study the biology of receptor by let's say applying one of these peptides to a mouse model, you don't know if it's actually an effect that you see via one receptor or multiple receptors. The small molecules that have been developed for this family of receptor are quite sparse. There's 2 well-characterized antagonists, RF9 and BIPP3226, which are non-selective for NPFF1 and 2. And in fact, the BIPP3226 is also an antagonist of the NPY receptor, demonstrating a further degree of non-selectivity. So I wanted to tackle this using the methods that I had just developed for receptor structure prediction to see, what can we gain by trying to predict and understand small molecule binding at these receptors. So I started with the same method, homology modeling of the NPFF1 receptor using the Rosetta methodology. I selected 3 templates that had pretty decent identity, all mid 30% identity to the NPFF receptor. And I got models that looked fairly well converged in the transmembrane region, and of course had high dynamics in the various loops. All what we expect at this point. But I did play one little trick that I hadn't previously done, in which I included the presence of a small molecule during the modeling step. And I was able to get away with this because of this BIP3226 compound shown here. It is highly related to this other ligand, URMK299, which was in a co-crystal structure with the Y1 receptor. And there's only one difference between the co-crystal ligand and the BIBP ligand, and that's the addition of this group here to the guanidinium. And I had this idea that if I just chop this bit off, I could steal the coordinates of the remainder of the ligand, place it in the bound state in my templates as I expect for the Y1 receptor, and then use that as a cofactor during the modeling to keep the pocket open. And in fact, that worked really well. The pocket not only stayed open, but side chains that are known to be important for interacting with this ligand and others in the series maintained the right rotameric state that are needed to then interact with the ligand, even in the models. And I then asked the question, okay, so for pre-optimized or not optimized models, what's the effect on actually being able to dock new ligands into these model pockets? And so for the pockets that had not been optimized, so those that didn't have the ligand in the system during model building, I was only able to dock on average 1 to maybe 2 known actives, but largely with the tail end towards the 0 to 2 range. However, in the optimized models, so including the ligand during the co-modeling process, there was an increase in the number of models that were able to dock a number of actives back into this pocket. And in fact, was getting a significant number of models that were able to get 5 or more actives. The numbers of actives here are quite low, as sort of highlighted by the fact that there just hasn't been much chemical development for this family of receptors. So I was stealing stuff from here, and the maximum would have been 20 for this family. But it clearly demonstrated by doing these tweaks, keeping the pocket open, pre-optimizing it for ligand recognition, you actually could then get a model of a receptor that was useful for ligand docking. And so that begged the question, can these optimized models be used prospectively in structure-based drug discovery? So at the time, as Nicholas mentioned in the introduction, I was working with Brian Schoiket at the University of California, San Francisco, and I had joined his lab primarily based on the success of his lab at doing large-scale structure-based virtual screening at GPCRs and at other targets. I mean, it seemed they were just continuously having success, and I wanted to get in on that success. And I thought, you just go in, take my model, dock, get a hit. That's what it is. But as I started working with Brian and the team, I realized that there's so much more to structure-based virtual screening than may at first appear. And so as I was sort of learning all these trials and tribulations myself, at the same time with another postdoc that joined the lab simultaneously, we decided to document all of this together to really demonstrate outward what goes into the structure-based virtual screening process. And basically the key takeaway was that the docking step is one of the smallest steps, and actually in terms of the time scale is one of the shortest steps of everything that you do in a structure-based virtual screen. There's so much work that goes into selection of the target and optimization of the target before you get into the docking, and then a significant amount of work that happens after the docking to get you down to a number of compounds that you might want to test, and then hopefully get real experimental data that demonstrates hits. And so we wrote this up a couple of years ago and highlighted a lot of the key decision points that have to be made in order to go into this, what I originally thought was this one step here. And so learning this process trial by fire myself, I then wanted to apply it to NPFF1R with the models that I had generated. So that's shown here, the models with the various dynamics here in the loop regions, but a very well converged binding pocket. Through a set of retrospective docking to select a pocket, a model that was going to be the one most likely to identify actives in a prospective setting. I carried that forward into a large-scale screen. So this is 177 million compounds that I docked primarily from the enamine real library. These were all monocations because I wanted a positive charge in my end ligand. At the time, the enamine real library was maybe 400 to 500 million compounds. If anybody's been keeping up with it, it's around 4 to 10 billion compounds now. So this potentially could be considered a small scale docking in today's current standards. But at the time, it really is quite a massive docking campaign. So through a series of various post-docking filters, I ended up with 33 that we carried forward into in vitro testing. And luckily, and it's still amazing that any of this works in my opinion, we were able to find a hit in compound 16, which is shown here in yellow, overlaid with the BIP3226 compound. And what I think is quite beautiful about this compound is whereas BIP3226 requires this large scaffold to really accommodate two interactions, this guanidinium and this hydrophobic phenol ring, the compound 16 just simply plopped down right on top of the two key pharmacophores and ignored everything else about the BIBP compound. We were able to carry forward this compound through a series of optimizations, going from a 300 nanomolar initial hit down to 22 nanomolar. And the activity of this final compound 56 is on par with the BIP3226 compound, but you'll see the molecular weight is half of that. So the ligand efficiency is much higher, specifically because I said it's hitting the key pharmacophores and only the key pharmacophores. And truly, if you find a hit, it's already incredible, but then to have it a biologic effect in a mouse model is a whole nother level. And we were all very happy to see that the administration of this compound to mice had an analgesic effect in a hot plate assay. So it had been documented to a certain extent that the RF amide receptors could be involved in attenuating opioid signaling, but as I mentioned, the only compounds available to test this were non-selective, and so we didn't have a very good compound here to really tease this apart. But we found truly an antagonist at NPFF1 is analgesic in this assay, and it is an analgesia that's additional to morphine. So it can actually boost the effects of morphine through the opiate signal pathway. And then through another set of experiments demonstrated that its activity is not dependent on the opioid receptor, it's fully independent. So if you knock out the opioid receptor in mice, morphine no longer has any effect compared to baseline, but compound 56 is still quite potent in the hot plate assay. And simultaneously for non-genetically modified mice, if you co-administer naloxone, an antagonist of the opioid receptor, you don't see any change to the efficacy of compound 56. So this paper is currently going through review, but it is available on bioRxiv. And it's one of these things that still blows me away that going from a primary sequence of a protein, we can predict a model, optimize it, use it in a prospective setting, and get not only an active hit compound, but an active in vivo compound. Now, NPFF1 might have been considered an easy case study because I mentioned I had that almost exact compound binding pose from the Y1 receptor co-crystal structure. So we wanted to test this on sort of a more likely scenario. And in this case, we chose the PAR4 receptor, specifically trying to find an antagonist. So there is no experimental structure of PAR4. There's only PAR1 and PAR2. And the bound ligands to PAR1 and PAR2 occupy distinct binding sites. So there's actually 3 different binding sites over the experimental structures that are available. So trying to identify even what binding site we might go for PAR4 was an open question. Then when we look at what is the ligands that are bound to PAR1 and PAR2 in their experimental structures, and how does that compare to PAR4 chemistry, we see that there's largely no overlap. So Vorapaxar here in green, this is the bound state of Vorapaxar. This is a PAR1 antagonist. And for PAR4, there's this BMS series, this imidazothiazone, that looks distinctly different from Vorapaxar. I mean, I wouldn't know how to align these two molecules just looking at the 2D structure on their own. So without the easy scenario like I did with NPFF1 of stealing ligand coordinates, what do we do here? And what we decided to do was actually to go ahead and build PAR4, build the PAR4 models exactly as I did with NPFF1. And we would actually use Vorapaxar, the green ligand, in the binding pocket. We thought BMS probably was going to bind in the primary orthosteric site. Vorapaxar was in the primary orthosteric site, so we thought, let's use Vorapaxar, put it in the model. We know it doesn't bind PAR4, but it at least keeps the pocket open. Then once we have models of PAR4 with an open pocket, we can align and dock BMS3 into the binding pocket and get models that start to look reasonable, and then re-optimize the binding pocket around the BMS3 pose that we had decided upon. And so this generated a number of different models at each step that then sort of propagated on top of each other. And all of the output models in the end all got challenged to the same retrospective control calculations that we described in our overview paper, to identify a single best model to carry forward into the prospective virtual screen. So here we did 164 million compounds against the PAR4 receptor, and selected that down to about 100 for experimental testing. And again, somehow it still is a bit strange, but we got hits. Going through this very complex system of really building a model around unrelated chemistry, we got hits that were active over a number of different signaling pathways through the PAR4 receptor. And these were able to be optimized with traditional medchem SAR by catalog type approaches to some potent hits around 100 nanomolar at PAR4. And in the end, I want to just show what the binding pose of compound 31, the predicted binding pose of compound 31 compared to BMS3 was, and just sort of reiterate the fact that we went from a PAR4 model bound to Vorapaxar to get the BMS pose, so already something distinct from Vorapaxar, to now get a pose of compound 31, which is again distinct from BMS3. And so it shows that being close in conformational space to the structure, at least having pretty accurate binding pocket side chains, allows a strong sampling of the conformational space for ligands in this area. And it's to me one of these things that it still excites me to this day to use these methods, homology modeling, so on and so forth, to get new chemistry for receptors that don't have structures. And so in the end, I want just a few takeaways here. And primarily, I hope that I've convinced you that structure prediction is a valid method for generating relevant structural models. And if anything, AlphaFold these days is hopefully convinced people even more of that. But both AlphaFold models and homology models really suffer from this pocket collapse problem. And the absence of a ligand during the co-folding stage generates pockets that you just cannot get ligands back into. And we've shown that, and a number of other people have now shown this for AlphaFold models, that ligands no longer can fit back into the pocket. There are, of course, new methods, the AlphaFold latest and Rosetta Fold all atom, that are now trying to do some co-folding together because they recognize that this is a problem, but they're quite new and to be honest, haven't been broadly tested. So it remains to be seen. And I think it's exciting to see where it goes next. But then, if you can include a ligand during the co-folding process, the optimization of that binding pocket allows you to go out and explore new chemistry and get new hit ID for compound or for receptors where you only know the primary sequence, which is, I think, it's just truly powerful to go from sequence to new chemistry. So with that, I think I will stop and take questions, and I hope you're just as excited as